My topic is prenatal diagnosis. The prenatal diagnosis or prenatal screening is testing for disease or condition in a fetus or embryo before it is born. There is a branch of medicine which is called a perinatology that is concerned with the well-being of the fetus and the newborn infant, generally covering the period from about 26 weeks after fertilization to 4 weeks after birth. The aim is to detect such a neural tube defect, Down syndrome, chromosome abnormalities, genetic disease, and other conditions such as spina bifida, Tay Sachs disease, sickle cell anemia, thalassemia, etc. There are three purposes of prenatal diagnosis. First one is to enable timely medical or surgical treatment of a condition before or after birth. Second one is to give the parents to chance to abort a fetus with the diagnosed condition. Third one is to give parents the chance to prepare psychologically, socially, financially, and medically for a baby with a health problem or a disability. Nowadays, different kinds of methods are used in prenatal diagnosis, such as amniocentesis, chorionic villus sampling or CVS, alpha fetoprotein assay or AFP, fetoscopy, percutaneous umbilical cord blood sampling, ultrasonography or USG, computerized tomography or CT, and magnetic resonance imaging. Next, we are going to study CVS or chorionic villus sampling. In CVS, it involves taking a sample of placenta, whereas in amniocentesis, it involves taking a sample of amniotic fluid and the fetus cells. Next is biopsies of chorionic villi may be obtained by inserting a needle with the help of ultrasonography through the mother's abdominal and uterine walls into the uterine cavity. The biopsies is a process involves the extraction of sample cells or tissue for examination to determine the presence or extent of a disease. Then this tissue is used for detecting chromosomal abnormalities, inborn errors of metabolism, and X-linked disorders. The major advantage of CVS over amniocentesis is that it allows the result of the chromosomal analysis to be available several weeks earlier than when performed by amniocentesis, because CVS can be performed between 8 to 10 weeks of gestation. The rate of fetal loss is 1%, slightly more than the risk from amniocentesis.